Welcome everyone. I'm Tracy Woodruff and I'm a professor in OBGYN and I'm the PI of our EaRTH Center. I think we've had some interest from people. We have a lot of interest from inside the university as well as outside the university. So welcome to everyone who is a member or affiliate, or if you haven't been to one of our webinar series, welcome also. So we're really excited to be kicking off our monthly webinar series with two of our key members, Dr. Josh Robinson and Dr. Hao Chen. And for just before we get started with their talks, I just want to let everyone know, as I mentioned, that we're going to be having a monthly webinar series through the EaRTH Center covering different topics and aspects of environmental chemical exposures and health outcomes with a focus on particularly on reproduction and development, which is why we're so excited to have Josh and Hao speak with us today. Also, just a reminder to people, you can follow us on social media channels and retweet any and all information that we have related to the center. And if you're a center member, please remember to cite the center as part of your excellent work, because it helps us continue to have the support that we have from the National Institute for Environmental Health Sciences, who funds our P30 Center at UCSF. So today, Josh and Hao are going to talk about environmental chemical exposures and mechanisms underlying human placental dysfunction and disease. So we're very excited about this talk because they're going to focus on their research as well as feature some of the work that we're doing at the center. And I believe Josh is going to go first, and then Hao, is that correct? Yep, they're nodding their heads. So Dr. Robinson is an assistant professor in the Center for Reproductive Sciences and in the Department of OBGYN here at UCSF, and he has been at UCSF since 2013 and joined the faculty a few years ago. And he runs a lab studying toxicological consequences of environmental chemical exposures during early pregnancy and their mechanistic links with developmental disease. Josh and I have collaborated on many different projects. He has a PhD in toxicology from University of Washington, and he trained at NIEHS and also in the Netherlands, just super interesting. And so he's going to give an overview of his group's research on using primary human placental cells and tissue models, systems biology, and in vivo biomarker approaches to studying the relationship between chemical exposures and human pregnancy complications. And the other person speaking today is Dr. Hao Chen, who is a postdoctoral fellow, and he works with Josh and Dr. Susan Fisher, who is also a member of our center. And he got his PhD in pharmacology and toxicology at UC Davis in 2017 and is a K99 awardee. So congratulations again, Hao. We're very excited about that. And he's going to talk about his work using proteomic-based approaches to understand the effects of environmental chemical exposures. And this is great because we're focusing on chemicals that we study at UCSF, PBDEs and PFAS exposures on human cytotrophoblast differentiation. So I'm going to turn it over to you guys.
Well, thank you for that wonderful introduction, Tracy. And of course, this wonderful opportunity to present.
Oh, Josh, I have to say one thing. People should put their, sorry, I like forgot my host duties, which is to please chat your questions, comments in the chat. We like to have a lively chat. We'll also take questions at the end so people can raise their hand. It's better if you raise at the end because I probably won't notice you right away. So sorry, I wanted to just say that in case, because I know when you're talking, Josh, something interesting will come up and people will want to talk about it in the chat. So that's why I had to stop and say that.
Of course, perfect. Well, thank you for this wonderful opportunity to discuss our research with all of you in this virtual setting and just be involved in this center with all these people who are so interested in environmental health questions. So as Tracy mentioned, today I'll give a brief overview of our work, which aims to determine how environmental exposures contribute to human placental dysfunction and developmental disease. And the outline of my presentation is as follows. I'll first give a brief background regarding environmental chemical exposures in human pregnancy. I'll provide a few examples of how we are using primary in vitro models to study the effects of environmental chemicals on human placental development. And lastly, I'll quickly discuss how we are leveraging these studies to investigate the links between chemical exposures and perturbations in placental development in humans. So we are interested in this relationship between chemical exposures and pregnancy due to several reasons, which I'm sure many people on this call share as well. As many of you know, the number and total production of chemicals at an alarming rate in the US and globally, outpacing the population. And the fact is that the majority of these chemicals that are being used in consumer products in the marketplace have very limited regulation. It's estimated that about 2% of chemicals that are registered by EPA have been thoroughly tested for their ability to cause developmental toxicity. As for human pregnancy, due to studies by Tracy and others, we know that many of these chemicals make their way to the embryo fetus, including suspected or known toxicants such as heavy metals, pesticides, flame retardants, and many other chemicals that we know really little about. And we're deeply concerned about environmental interactions during pregnancy because we know that little effects by toxic chemicals can have dramatic consequences during gestation that manifest in childhood and adult life. So as mentioned, our group is particularly interested in the impact of chemicals on placental development and function and how these impacts underlie pregnancy complications and alterations in development. And there's a growing body of evidence suggesting that chemicals contribute to pregnancy complications and placental toxicity due to a growing body of work in epidemiology and toxicology. To understand these relationships, we have to understand what makes the placenta a vulnerable target. The placenta is a critical organ for embryonic and fetal development, undergoes dynamic changes during pregnancy to serve the baby's needs. It acts as the physical connection between the maternal and fetal units. It serves as the master regulatory barrier involved in the transfer of oxygen, nutrients, and many other molecules. It removes waste products. It serves as the immunological barrier and also serves as an endocrine organ, producing key hormones for pregnancy. Unsurprisingly, due to the many roles that it plays, successful placental development is strongly tied with successful pregnancy and the development of the embryo and fetus. The chorionic villi serve as a key functional unit of the placenta, rapidly transforming from primary to complex structures over the 40 weeks of pregnancy to increase surface area to enable exchange of nutrients, waste, and gas between the maternal and fetal blood supplies. And the associated trophoblasts, specifically the syncytial trophoblasts, which are multinucleated hormone-producing cell, and the cytotrophoblasts form a bilayer during early and mid-pregnancy, which create this barrier between the maternal and fetal blood supplies and facilitate the exchange of nutrients, waste, and gas between the mom and the baby. Chorionic villi, which attach to the uterus, also known as anchoring villi, also play a different role. These cell CTB or cytotrophoblast-rich cell columns provide a gateway for cytotrophoblasts to invade into the uterine wall and to remodel maternal blood vessels, which increase elasticity and increase blood flow to the intervillous space, which again help this exchange of nutrients, waste, and gas between the mom and the baby as the baby grows and demands more of nutrients, waste, and gas exchange. So unsurprisingly, due to their critical roles in development, many of the common pregnancy complications that afflict a large proportion of the population, including preterm birth, preeclampsia, fetal growth restriction, and accreta, are associated with a variety of pathologies involving impaired cytotrophoblast invasion or defects or stress in the syncytial trophoblast layer. Therefore, our group really focuses on these different aspects in trying to connect how environmental chemicals affect the placenta and how this leads to pregnancy complications and changes in development. And this hypothesis that chemicals impact the chorionic villi and the associated trophoblasts is really supported by a wealth of evidence in rodent and cell models, suggesting that chemicals induce stress and alter trophoblast function, even though the mechanisms are still undefined. So our group, uniquely, we have access to placentas from all different ages, including first and second trimester and term. by working with Susan Fisher's group. And we use primary human placental cell tissue trophoblast models to study these environmental interactions. Specifically, we use two models, one being the primary villus explant, which, as you can tell, is essentially a fragment of the chorionic villi, as well as primary villus cytotrophoblasts, which, as you can see in the middle here, involves kind of a complicated procedure to take placental tissues and isolate these cells. And then we can use these cells to study toxicological interactions. These models have been used to study aspects of placental biology and disease for at least 3 decades, while their potential application in toxicology is relatively unknown. However, we feel that these models have many advantages. They retain in vivo characteristics. So for example, the primary villus explants maintain the cytoarchitecture that they have in vivo. And the primary villus cytotrophoblasts that we isolate emulate their invasive phenotypes in vivo, and we can study sort of these interactions in the in vitro condition. Other advantages include the direct relevance. We're obviously studying human cells and trying to understand interactions that occur in humans. And humans, as you know, are not equivalents to rodents, and cell lines have a lot of differences in terms of their in vivo counterparts. And something unique about these models, we can study the effects of chemicals across different gestational ages. So we can look at the susceptibility of these chemicals or the vulnerability of these cells to a variety of chemicals early in pregnancy as well as at term. Another advantage, which also can be a disadvantage in some aspects, is these cells are genetically unique, so we can generalize our results in a much different way. And also, placenta, because it is a large organ and has many different cells, is actually a robust model in terms that we can do, we can look at multiple groups in the same experiment. So as I mentioned, isolated cytotrophoblasts differentiate and invade into this phenotype that's similar to what we believe occurs in vivo. So they do not proliferate, but that doesn't mean that they're not highly active. In culture, as you can tell within the first couple days, they form cell aggregates. They throw out many different cellular processes. They undergo remodeling. They almost emulate what we believe is kind of the formation of the cell columns of the anchoring villi. And they also release a ton of microvesicles and exosomes that are involved in cell communication. Not only have we produced several different types of movies, which we can look at for many hours because they're quite exciting to stare at, but we've also looked at the dynamics on the molecular level. So this is just some examples of some of the work that we've done looking at gene expression. So the figure here on the left is showing over 2,000 genes that we know significantly change over time within the first 2 days in culture. And maybe more importantly, the genes that we see change are related to these pathways that we know are critical for cytotrophoblast differentiation and invasion, including vasculature development, anatomical morphogenesis, and cell migration. Likewise, we know that a lot of the molecules that are really critical for invasion into the uterine wall are being upregulated during this time. Again, just confirming that we are looking at this invasive phenotype. Likewise, a downregulation of genes and markers which promotes syncytialization, which is another route of CTB differentiation. And more recently, just to kind of give a leeway into Hao's work, we began to look at this on the proteomic level. So a lot of these studies have certain the baseline characteristics. And again, this is added on top of decades of work by Susan Fisher's group, as well as others using these models. We began to think about how we can use this for toxicology. So for that, it's been to define a battery of testing endpoints with functional and clinical relevance that we can analyze. So this includes looking at things like cytotoxicity, apoptosis, oxidative stress, as well as functional endpoints that are relevant for placental biology, but also disease, including migration and invasion, HCG production, and villi integrity, as well as molecular effects which are relevant, such as inflammation and stress, hormone signaling, and differentiation. So using these battery of assays and these models, we've gone forward to study the effects of chemicals to see if we can use these model systems to connect the links between chemical exposure, changes in placental development, and pregnancy complications. For today, we're really going to just highlight our work with flame retardants, but I do want to mention that we have several ongoing studies looking at a variety of different compounds in our group, in case there's other collaborators here on the phone who are interested. So today we'll primarily talk about flame retardants. So the initial studies that we worked out in using these model systems really focus on PBDEs, also known as polybromine diphenyl ethers, which, as many of you know, are widely used as flame retardants, even though over the past decade, they've been phased out in the US. Due to their persistent and ubiquitous nature, these chemicals are still, though, of concern. They're found in the majority of humans still to this day, and they're found in cord blood and fetal tissues, including the placenta. There's 209 different congeners based off the placement of the bromine atoms of these structures, with BD47 being the most common species found in human. And while these chemicals have been phased out due to their potential to cause developmental toxicity, and specifically developmental neurotoxicity being one of the most sensitive endpoints, in the last 5 years, there's been a variety of evidence that's came out in humans and animal models suggesting that these chemicals are placental toxic as well. So in our studies, as sort of a proof of principle to use these models and to study these chemical effects, we took primary human villus CTBs from second trimester placentas, and we looked at how these chemicals affect a variety of endpoints in a concentration-dependent manner, specifically looking at BD47 as well as BD99, another common congener. And we looked at endpoints related to cytotoxicity, invasion, migration, and also looking at the transcriptome. And these are just some examples of some of our results related to cytotoxicity. So we found that both BD47 and BD99 induce cytotoxicity in primary human CTBs after a 24-hour exposure duration. Hopefully you can see on the top left here, we saw a reduction with BD47, BD99 in terms of cell viability, with significant effects with 10 and 25 micromolar. And on the right, we saw an increase in LDH activity, lactate dehydrogenase, which is an enzyme that we test as a proxy of cell death. And hopefully you can see this inverse relationship with significant effects occurring with 10 and 25 micromolar. Using benchmark concentration modeling, we looked at equipotent concentrations reflective of a 50% decrease in viability or 100% increase in LDH. And here you can see on the bottom left here, just examples with other endocrine disruptors and contaminants of concern, such as BPA and PFOA, just showing that these compounds, PBDs, are relatively toxic in this model system. And hopefully what you saw, we've also done several movies to kind of look at the time response of cytotoxicity. We do see effects occurring as early as 4 hours. So of course, cytotoxicity is a very interesting endpoint, but maybe not as physiologically relevant. So we decided to look more at concentrations that are subcytotoxic. And the first thing we looked at was the toxicokinetics to really understand if BD47 is taken up by these cells, even at concentrations lower than 10 micromolar. So in this study, which was fairly simple, we looked at BD47 in either the cells or the media after a 24-hour exposure. And hopefully what you can see here on the left is that after 24 hours, BD47 accumulated in the cells with 1 and 5 micromolar. What was very interesting is this relationship was nonlinear in terms of the fraction that ended up in the cells. So with 5 micromolar, we saw a much larger proportion of the compound being accumulated in the cells. And this really supports the nonlinear molecular response, which I'll show in the following slides between these two concentrations. Another side note, which I left out of this slide, is that these compounds were not metabolized in the CTB model. And we do believe this is due to a lack of CYP2B6, which is the main CYP P450 enzyme involved in breakdown of PBDs. Looking at these concentrations further, we asked whether this affects some of the functional attributes of these trophoblasts. And we found that in a concentration-dependent manner that BD47 inhibited CTB migration as well as invasion. And these studies were in line with studies in human and rodent neural cell lines, as well as more recent studies in placental cell lines indicating that PBDs alter cell migration. Again, a toxic concentration of 1 micromolar, we studied the transcriptome to really understand what are the mechanisms in which these compounds might be disrupting function. And hopefully what you can see on the left, which is essentially the effects of 276 genes by BD47 after a 24-hour exposure duration. And this study, we added some complexity in that we decided we would either expose them after they initially ablated at 3 hours or after they form these aggregates at 15 hours. And hopefully what you can see, independent of kind of when the exposure occurred, the 24-hour exposure induced a similar change in these genes in terms of their fold change response. Maybe more interestingly is what are these genes and what are they associated with? If we run an enrichment analysis, we saw an overrepresentation of biological processes related to placental development or chemical stress, suggesting that particular pathways such as morphogenesis, vasculature development, cell migration were being impacted by BD47, as well as pathways related to inflammation and lipid and sterile metabolism. For this study, we decided to focus on those particular pathways to look at particular molecules that may be of interest in relation to placental development and stress. And these included some key proteinases, MMP1 and MMP8, which are involved in the breakdown of the extracellular matrix and really key for CTB invasion and have been shown to be linked with placental diseases such as preeclampsia, as well as IL-6, which is a major cytokine involved in inflammatory response and has links with preeclampsia as well as neurodevelopmental diseases such as autism. Using these targets, we decided to do QRT-PCR validation to really dive into the concentration-dependent response in relation to these different molecules highlighted here on the left, as well as a few others. And hopefully what you can see is kind of the dramatic changes that occurred with really 1 micromolar serving as the point of departure in these studies, where with 5 and 10 micromolar, we had much more significant effects, and 1 being significant, but of lower magnitude, where concentrations which are more relevant, probably physiologically, we saw no significant changes. So this really set the standard in terms of a proof of principle to then begin to look at other chemicals. Kind of a natural transition was then to look at the alternative flame retardants, which are serving as replacements for PBDs as PBDs have been phased out. I think this is a particular area of concern. AFRs are increasingly being introduced into consumer products, and there is very limited information regarding their health effects. And what I think is very worrisome is this area is very complex. Before we had polybrominated diphenyl ethers, which represented kind of this class of compounds. Now we have multiple classes, and currently there's about 50 different flame retardants that are being used with many diverse structures. What's kind of scary is already these chemicals are widely detected in the environment in humans, and the degree of exposures are similar or exceed the levels of PBDs that we saw previously. And while many of these chemicals may have shorter half-lives, these results suggest that we're being faced with maybe more continuous exposures of these chemicals. Initial studies in vitro suggest that they do differ in potency, and that a subset are as toxic or more toxic as PBDEs. More recently, recent studies have shown that in rodent and in vitro, that many of these compounds might be linked with placental toxicity. So in our studies, we were able to receive a panel of different alternative flame retardants from Mom Tabell at NIEHS, who's been in these collaborations to really look at a collection of common flame retardants that are being used in consumer products to study their effects in our CTB model. So initially, as we did with the PBDEs, we looked at cytotoxicity, looking at viability and cell death using this LDH activity assay. We modeled the concentration responses and determined benchmark concentrations. And we used this information to prioritize specific compounds that we found that are toxic in our study to then look at the transcriptome to identify genes and pathways that may be disrupted by AFR exposures. So I'll just briefly go through this due to time, but what we found is that AFRs decrease cell viability and cause cell death, very similar to BD47. Only one compound out of the panel, which was TSEP, was found to not be toxic within a concentration of 0.03 to 30 micromolar. And in general, these chemicals were similar in terms of their potency, within 1 to 2 orders of magnitude when after doing benchmark concentration analyses. And actual specific AFRs, such as TBPA, TMPP, IPP, and Firemaster 550, were shown to be as toxic as BD47. And this figure here shows the range in benchmark concentrations. Hopefully you can see that the two assays were relatively comparable. And in terms of BD47, you can see that these compounds here are as toxic or even more toxic when looking at equipotency. What the study also showed is that specific organophosphates, for example, the chlorinated OPs, seem to be a little less toxic in our system. So following up with these analyses, using borderline cytotoxic concentrations, kind of the max in which we want to study, we looked at the changes in expression that were associated with exposure. And we looked at 5 particular compounds, including BD47, for these alternatives. And we found that TBPA and IPP were the most potent at disrupting gene expression. So a question for us is, do these chemicals act via similar mechanisms? And what's kind of concerning is in our first examination of some of these gene expression changes. So we first looked at here with BD47, IPP, and TBPA, IPP being a mixture of organophosphorus flame retardants and TBPA, which is a common bromophenol that's used as flame retardant. You can see if we look at the 48 genes that were dysregulated and we see in a heat map, even without knowing what they are, you can hopefully see that these are commonly altered in the same direction. So BD47, TBPA, and IPP are upregulating specific genes and downregulating. And when we look at the specific functions, some of these are very interesting related to endocrine function as well as pregnancy complications such as CRH, HSD11 beta 2, and ESRRG. So what these types of studies have created is an understanding of potentially how these chemicals contribute to placental toxicity, proposing specific pathways related to differentiation, migration, inflammation, sterile metabolism that are disrupted, and as well as functional deficits related to migration and invasion, which may underlie trophoblast dysfunction and changes in placental growth as well as disease. These studies also are comparable to some of the work that we've been doing with our neurodevelopmental systems, developing kind of a holistic view in how these chemicals may disrupt pregnancy, as well as informing different researchers, including epidemiologists, people interested in biomonitoring of these chemicals, as well as biomarker analyses. And one example that we have internally was based out of the UCSF Peak Children's Center, led by Tracy Woodruff, where we use this information and leverage this information that we found in vitro, then look at clinically if these relationships exist. So in this analysis, what we did is we looked at 17 different PBDEs in a variety of different matrices, including placenta and maternal and cord sera. And we looked at placental morphology, specifically looking at hallmarks of stress and development, including fibrinoid deposition, leukocyte infiltration, and uterine blood vessel modification by the CTBS, as well as protein expression of specific biomarkers that Susan Fisher and many others have shown are really key for trophoblast invasion, including integrin alpha-1, cadherin 5, also known as V-cadherin, and matrix metalloproteinase 1. So in these studies, what we found is that, as I mentioned in one of the first slides, PBDs were found in the majority of women and fetal tissues, including the placenta. So BD47 was found above level of detection, all the different tissues. And we looked at a subset of these samples, and we looked at integrin alpha-1 staining. This first figure here just shows you, irrespective of the exposure or the chemical level in the sample, it's just showing the immunoreactivity of the CTBs as CTBs invade. So this is CTBs within the floating villus, anchoring villus, proximal, distal, CTB interstitially in the uterus, as well as CTBs in the endovascular or remodeling the blood vessels. Hopefully you can see that the typical patterning of this ITGA1 molecule shows increased presence as the cells invade. Here in B just shows that patterning. So here, these are CTBs that have invaded into a maternal artery. And here you can see the high expression of ITGA1. And here is the association with PBDE levels and ITGA1 immunoreactivity and endovascular CTBs. So what we saw is a negative correlation, essentially, with the higher levels of PBDEs being inversely correlated with ITGA1 immunoreactivity, suggesting that chemical levels are negatively associated with this molecule's expression in these key populations of cells important for placental development. Okay, so in summary, hopefully I've shown that there's a growing concern that environmental chemicals cause placental toxicity and contribute to pregnancy complications and developmental disease. Chorionic villi and associated trophoblasts may be susceptible targets of chemical toxicity. And hopefully I've shown you that in our studies, chemicals alter trophoblast function and gene pathways related to stress and development. In ways that we can translate this information, we show that PBDE levels in women are associated with altered profiles of CTB proteins involved in uterine invasion and endovascular remodeling. And in summary, a lot more to be done. Our results suggest that chemical exposures contribute to trophoblast dysfunction and human pregnancy complications of placental origin. So we feel that these studies are really just the beginning. There's definitely several directions that we'd like to take this, and we're definitely looking for partners and how to go forward with this. One is to enhance our primary CTB tissue models for chemical screening, so streamlining the assessments to perform high throughput analyses and include multiple compounds to analyze the large chemical space that we're facing. To identify molecular signatures correlated with placental toxicity and pregnancy complications, I shed light on some of the transcriptomic analyses that we're doing with some of these chemicals. We now have data with heavy metals, perfluorinated compounds, as well as pesticides. So we're very interested in kind of going forward with these studies. We're also interested in the application of single cell or cell capture technologies to elucidate the sensitivity of specific cell populations to chemical agents. And finally, this is a nice gateway to Hao's work, is the use of proteomic and genetic functional approaches to delineate common and unique toxicity pathways of trophoblast dysfunction. So with that, I want to say thank you to everyone on the call and this wonderful opportunity by the UCSF EaRTH Center to present our work. And specifically, I want to highlight members of my lab, Hao Chen, who's been just a wonderful postdoc working with me for the last couple of years and has really moved these projects moving forward. Susan Fisher and her incredible team of placental biologists who've provided such an amazing knowledge base, and of course, initiated these studies with Tracy as part of the Peak Children's Center. Tracy Woodruff and her awesome team at PRE, who I could fill this whole slide with all their mentions, and specifically Julia Varshavsky, who's really pushed forward these translational studies that I briefly described. And lastly, Junsu Park and his group at DTSC Cal APA, which have done a ton of this biomonitoring work that really serves as the foundation of these studies. So with that, I'll say thank you and pass it on to Hao, or take questions at the end. Or am I taking questions at the end or no? I don't know.
Yeah, we'll do questions at the end. So we can transition the slides.
Thank you very much.
Yeah, thanks, Josh. It's always amazing to hear you talk. And I love your videos. Can't get enough of those videos.
We need more videos.
If I was on TikTok, I'm sure I'd see your placenta video on TikTok. You can tell me the song you're going to pick. It's not driver's license. Anyway, so I'm going to turn it over to Hao.
Sorry, I was on mute for a second there. What I just wanted to say is I think Josh obviously introduced everything really well. And what I said that I think is most relevant is that we really are kind of at the beginning here of trying to understand what's going on. I like to show this slide in general because I think it reminds of how interdisciplinary the work it is that we're trying to do, which is that we need to rely on basic biologists, epidemiologists, environmental activists, policymakers, clinicians everywhere to kind of broaden our understanding of the basic biology that might be affected by environmental chemicals, linking those environmental chemical perturbations to adverse outcomes. And from there, by understanding what's going on and fully addressing uncertainty, can we really have an impact on public health at a significant level? And I think this is kind of the strength of the EaRTH Center, and which is that is a truly interdisciplinary effort to kind of expedite what we know about environmental exposures and how they affect reproductive and developmental health. And as part of the EaRTH Bioassay Facility Core, there's many different ways to approach these questions, which I present here in kind of a simplified way of what the Bioassay Facility Core does. One is just saying what sort of chemical exposures exist in environmental and biological matrices, with again, the work being done by Dr. Jun-Soo Park. The other is what sort of chemical exposures can cause toxicity via high throughput chemical screening methods done with Dr. Jennifer Fung. Another methodology that we do, and as I like to highlight, is what sort of proteins are impacted by chemical exposures. And here it's done with under the mass spec lab here at UCSF. And I'll talk a little bit about what that entails and some of the work that's already been done as part of the EaRTH Center related to this. So when we talk about mass spectrometry and proteomics, we had the simplified notion of a cell in the textbook. But what actually is occurring in the cell is that there's many, many proteins there, and they're all dynamically changing. So when you had that old figure where it's like kind of mostly empty space and a lot of cytoplasm, and there's like one mitochondria there, it's not fully representative of what's actually going on in the cell or any particular biological matrices that you're interested in looking at. There's actually a lot of things going on. And ways to kind of understand or approach what's occurring in the general proteome, or all the proteins that are occurring within the cell, is to use these mass spectrometric approaches. And I'll talk very, very briefly in a very simplified manner of what mass spectrometry kind of does. And so as many of you guys were locked down a whole last year, some of you guys probably took up cooking or baking and those kind of things. And when you think of a general biological matrix, you can think of it kind of like an emulsion, right? Like salad dressing or mayo. Like there's fat components and there's water components, and they're all mixed together. And it's a much more complicated idea than that. But what mass spec is essentially trying to do is we're trying to separate all of those very complicated components, separating those things that make them together, and feeding it into the mass spec, which is essentially a giant kitchen scale. It just measures the mass of objects, like it's suggesting. And from there, you can get an idea of what are the things that compose the matrix that you're looking at. This particular cartoon is just showing that you have a matrix or a sample, you inject it via various chromatographic methods. You can separate your matrix into simplified molecules based on their physical characteristics. A voltage is applied that ionizes your sample, and that gaseous ion is what gets fed into the mass spec and is what's actually being measured. And from there, you can determine what is composing your sample. What's revolutionary, or what's not revolutionary, what's advanced about the facilities we have here at the EaRTH Center and the type of work we do is we use a new workflow called SWAS-MS. The old methodologies of mass spec, as you can just see from the cartoon, one of the things that you're kind of beholden to is what is the first thing hitting the mass spec and how much of a certain molecule is hitting the mass spec, because that can change what kind of reading you get. And so what happened with SWAS-MS methodology, they said, okay, we don't really care about what's gonna get hit first, the mass spec. We're just gonna look at everything at once via various SWAS windows. And then from there, you can get a whole chromatogram of all of the existing ions within a particular species, a biological species. And then you can compare it to a spectral library generated based on your existing samples. And you can continually refer to that library spectrum that you've generated. This is superior and in many ways complements previous methodologies because it really increases reproducibility of your data. Because as I mentioned, in the old methodologies, you're really reliant on kind of a stochastic method of whatever gets to the mass spec and what's most intensely hitting the mass spec, determining what you actually measure. Via SWAS-MS, because you're basically sampling everything and you're referring back to a library that you've generated, you get really reproducible data. And that's very important for the type of studies we do, because we're doing comparisons of protein abundances across different samples. As Josh had alluded to, one of the things we look at is the cytotrophoblasts. So this is just a cartoon of an unremodeled maternal spiral artery. What happens is that these trophoblasts come in, they remodel it, they lower the resistance, and they increase the elasticity of the blood vessel to permit increased blood flow to the developing fetus and the placenta. As you might imagine, if you disrupt this process, it has various implications in things like disease. So in normal placentation, you have sufficient invasion of these cytotrophoblasts to the spiral arteries to remodel them. In abnormal placentation, there's either, in this case, shallow invasion of these cytotrophoblasts, or in some cases, there's over-invasion of these cytotrophoblasts. Ultimately, what that results in is a disruption in blood flow to the fetus and naturally can affect both the health of the baby and also the mother. And as Josh had mentioned, we utilize these cytotrophoblasts derived from the placenta to study exactly what is going on with these cells in terms of their context, in the context of disease. And again, as I mentioned, what is actually fully understudied and what we don't know is what these cells are doing in the context of environmental health. As many of you know, the placenta, in toxicological terms, is generally thought of as primarily a metabolic intermediate, right? It's almost like a giant liver barrier, blood-brain barrier thing. And so people are always interested, okay, is this thing getting past the placenta? And I think it's more appreciated now that the placenta is actually a target of exposures. And that's kind of the work that we're doing. It's understanding how is the placenta affected by environmental exposures. So we understand a lot, as Josh mentioned, based on the context of disease, but we're also trying to understand how environmental effects on the placenta can confer or increase the risk of disease on a public health standpoint. So here's just kind of a sample workflow that you can do via the SWAS-MS proteomics. In our case, what we did is we just cultured these cells, as Josh had alluded to, at various time points. From there, you can lyse these cells, you can extract their proteins, you chop them up with enzymatic digestion to make it easier for the mass spec to handle. You perform a sample cleanup to remove any salts or any other contributing factors to the mass spec. Then from there, you do, as I mentioned, this initial shotgun proteomics to derive a spectral library that you continually refer to via your SWAS runs. So then when your SWAS runs, you're basically sampling all of the ions in any particular biological sample that you can continually refer back to. So again, you have this data extraction, it will refer back to the original library you created from all of your samples. And then from there, you can derive relative protein abundances related to each individual sample that you're investigating. So this work has already been recently submitted. So we were just looking at baseline characteristics of the cytotrophoblast model from a proteomic standpoint. 3,000 proteins were assigned. 400 were shown to be differentially expressed as a function of time. And via bioinformatic workflows, you kind of just get a general idea, okay, like you have 3,000 proteins, what's actually going on? What are their dynamics? This is just a graph just showing that you can separate the differentially expressed proteins into 4 various clusters that have unique characteristics. So cluster 1 and cluster 2 are showing that proteins in these clusters are generally being upregulated as these cells are differentiating into an invasive phenotype. Either they have an immediate and sustained upregulation, or there's a steady climb. And likewise, there's 2 other clusters that show downregulation. Either there's an immediate and sustained downregulation, or there's an abrupt downregulation later on in culture. And each of these clusters has broadly a distinct gene ontology pathway process associated with each cluster, excuse me. So if you look at it by individual cluster, you can say, okay, well, in cluster 1, this expression profile seems to predict proteins associated with something like oxidative phosphorylation. Or if you're looking at a protein that's part of cluster 2, which it might be associated with expression profile related to cell-cell adhesion or cell migration. And from there, you can get a lot of idea of what's actually occurring in this very complicated mixture of proteins that you have in a type of model that you're looking at. You can furthermore look at it and stratify it by time. So by doing these methods, you can look, okay, each individual time point has a specific characteristic associated with these proteins in that time point. And then if you look at, say, specifically the most robustly changing proteins, you can also identify pathways that you would associate with your cell model. In this case, we see things we would expect from an invasive cytoplasm population, which is molecules are primarily associated with things like the degradation of extracellular matrix. And here's a list on the side of what kind of proteins that we can generally look at. And again, if you're looking at it from an environmental health standpoint, or even from a basic biological standpoint, that list that you get of proteins that are changing helps you get an idea of proteins that probably haven't been looked at very closely before. So in our case, we picked out some proteins that we've seen before, we haven't been looked at very closely before. One is this nitrogen 1, which is a composition of extracellular matrix. And as Josh has shown before, and like kind of all of us have, Josh and I have shown in kind of a cartoon form, you can look at it in kind of a actual physiologically relevant form. So this is just a slice of a placenta. And what you can see is, again, nitrogen 1 is a component of extracellular basement membrane. And the red stained cytokeratin 7 marks trophoblasts. What you can see is that these cells are really breaking down all the surrounding area of where they're actually implanting into the decidua. And furthermore, as they invade and break down basement membrane further into the mother, they're doing it in a very specific, highly sophisticated manner where they're basically surrounding themselves only, or they're breaking down only locally in the areas that they're invading. Furthermore, we can look at proteins that have been studied, like again, sequestosome 1, which is this marker for, or not a marker, it's associated with autophagy. And we can say that these cytotrophs are actually very robustly expressing this molecule. So from that understanding of a basic biological model, you can start looking at it in environmental health context. So as Josh had alluded to, what we did is we looked at it by exposing these cells to polyvalent diphenyl ethers, or PBDE47. Well, we saw that 200 proteins were differentially expressed as a function of PBDE47 treatment. These heat maps are just to illustrate kind of a general idea of what happens at each time point in terms of the proteins that are upregulated and downregulated. And what was surprising to us is that there was actually a very minimal overlap of differentially expressed proteins compared to the control model I just shown earlier. Why that's interesting is because, as Josh had shown, on a functional standpoint, that PBDEs can affect the ability of these cells to migrate or invade. So in that, intuitively, you would have thought, okay, if it's affecting that, it would affect similar pathways or similar molecules. And in fact, it doesn't. So there's actually only about a 4% overlap on the proteomic level. If you're looking at enrichment, or to look at the type of pathways that are associated with the proteins that are being affected by PBDE47 treatment, we generally see enrichment of pathways associated with exocytosis, but also cell-cell adhesion. And again, which is what you kind of intuitively would have expected. You can look at these proteins that are affected via stratifying at each individual time point and each individual concentration. And what you can immediately appreciate, these are just a dot plot showing each of these proteins, how much they change by fold change and how significant these changes are, with the bottom here being in the concentration of PBDEs, and the top here being listing each of the time points we looked at. You can see there's a clear concentration dependence. That is, depending on what concentration you look at, there might be minimal overlap or some overlap at each protein you're looking at. And there's very clearly a time dependence, which is at 15 hours and 24 hours and 39 hours. There's more and more proteins that are being significantly affected by a chemical treatment, which again, intuitively makes sense because these cells are seeing the chemical for longer and longer as they are being cultured or differentiated. From that, you can do a network analysis and identify proteins that might be changing and might be interesting to look at. So in our case, we looked at this NF-kappa B1, which is associated with oxidative stress. what we saw is that this CyFIP1 or cytoplasmic FMR1 interacting protein was being very robustly upregulated at 15-hour, but returned kind of normal at 39-hour. And this protein has never been studied in any context related to placenta, and is because it's primarily thought of being as a neuroprotein, right? FMR1 is related to Fragile X mental retardation protein. And as we all know, PBDs are primarily associated with neurodevelopmental consequences. And so what we've seen is that, okay, now we can look at these proteins and we can see that CyFIP1 is in fact expressed in these cells and expressed in the placenta. And as of yet, no one's really looked at what it's actually doing there. And so I think that's what's really exciting about doing this kind of work, is we're not just identifying how environmental exposures affect the public health standpoint, but also in a basic biological standpoint, there's many, many molecules that haven't been fully looked at in the context of just general health. And finally, as Josh had mentioned a little bit, one of the components of my K99 is to look at perfluoroalkylated alkylated substances. And I've generated currently, all of these samples have been collected where I've exposed these cells to PFOA, GenX, PFOS, and PFNA. I'll just present a little bit of work that I had done with the PFOA. So when I expose these cells to PFOA and do the same SWOS-MS workflow, what I see is about 200 proteins of 2000-hour detector are significantly changed at 24-hour. And specifically that migratory and adhesive processes are very overrepresented in these 200 proteins. And does that make sense from a functional point of view? And so as Josh mentioned, you can do these transwell assays where you culture these cells on a little insert, and these cells can basically invade or break down this insert, the membrane in this insert, to kind of invade to the other side. And what I had seen is that with PFOA treatment, it very robustly and significantly increases CTB invasion. Again, reinforcing that this class of chemical is not only having changes on the global proteome, but is clearly having a functional effect on these cells. And so what I hope I've conveyed is that mass spec-based proteomic approaches are a really powerful tool for identifying biomolecules affected by environmental exposures. And these environmental exposures can significantly affect the global proteome side of trophoblast, which can impact placental development and likely increases the risk of adverse pregnancy outcomes. And ultimately what we're trying to do here, obviously, as we're all part of the EaRTH Center, is to improve our understanding of environmental impacts, in our case, specifically at the placenta, or for whatever model you're looking at, to really improve public health awareness. And what I hope we can all do is just via the bioassay core and through all of the collaborations, just really kind of get a general and better idea of how these environmental exposures affect reproductive and developmental health. So again, with that, I would like to thank Dr. Susan Fisher and Dr. Josh Robinson, my K99 mentorship group, which is, again, Dr. Tracy Woodruff and Dr. Jujis, who are all here. I want to thank all of you guys for your support. And obviously, I would like to thank the EaRTH Center and all the placenta donors that make this work possible.
All right, thank you. Those were terrific, though how you didn't tell us what you found for Gen X, so we'll just have to keep waiting.
That will be the next one, yep.
Oh, no. Well, that was sort of a hint, but okay. So that was great. I was really demonstrated some really fascinating work that you guys have been doing. I know we're kind of close on time. So I don't know, Linda, you've been doing some chats. Linda Birnbaum, I see you're on here doing some chats in the chatter thing. Do you want to say something?
Well, I think both talks were fabulous and really very exciting. I think obviously I'm interested in some of the newer work with PFAS that I think Josh talked about first. And, I assume that hopefully he's talking to Rebecca Fry and seeing what her group is doing, because they're also looking at impacts of PFAS on the placenta and stuff, and I think several other things as well. So there may be some real nice opportunities for some cross-center collaborations going on there. But I just made the comment that TBBPA is such an amazingly high volume. It's the highest volume bromination retardant, and basically been ignored because it used to be used almost exclusively in a reactive mode, so it wasn't escaping. Well, now it's being used in more and more in an additive mode, and we're finding it more and more when people look in blood and in breast milk. So I think this, and again, its toxicity flew under the radar. It is a carcinogen. And there are now a number of other studies beginning to show a whole raft of effects. And let's see, what was the other comment I made on, the PBDEs? Clearly, and all these compounds are developmental neurotoxicants or in animal studies for sure. And some of them we have a lot of human data, but I think we should remember the PBDs are also really potent rodent carcinogens, both rats and mice and both males and females. So, you kind of can pick your target organ. But
Well, I was going to say, it was very interesting how when you talked about the protein that was linked to you, like people didn't really look at it because it was linked with neurodevelopment, right? Is that what you were saying? But now it's like, oh, maybe it's also a placentagen. Anyway, you can see that these pathways can go in different directions in terms of what we measure apically.
Exactly.
nice job.
Yeah, and I
Thank
just
you,
want to
Linda.
also put another plug in for this idea about, because you kind of basically did this scan, right? You're like, instead of picking what to look for, you're scanning what might be important, and that helps us elucidate these new mechanisms, which I think is really fascinating because we're doing a similar thing with the exposure work. So I think it really helps us understand better what's going on with biology. Anyway, does anyone else have any other comments or questions? All right, well, thank you everyone for joining us.